Hello, dearest friends. Now is my last lecture of my third week. Is the 15th lecture. I'm talking about enzyme-linked immunosorbent assay (ELISA). It's very specific technique for to detect the protein in the solutions, and it has very high sensitivity. Myself, Mehdi Hayas Shahi. I'm assistant professor at Interdisciplinary Brain Research Center, Faculty of Medicine, Aligarh University, Aligarh, Uttar Pradesh. The content of my slides is introductions and history of ELISA, basic immunological principles, what are the key components and the reagents, and also we discuss about type of ELISA, that is direct ELISA, then we have indirect ELISA, the sandwich ELISA, competitive ELISA, and material and equipment. Then we discuss a step-by-step protocol, then quantification and a standard curve, and then application in research and diagnostics. Advantage and limitations, finally summary and references. Introduction and history. ELISA enzyme linked immunosorbent assay. It is a plate based immunoassay technique that uses antibodies and enzyme mediated color changes. That's important. Color changes to detect and quantify protein, peptides, hormones, antibodies, and antigen in a biological system. That's very significant. In 1960, Allo and Burson developed radioimmunoassay, that's called RIA. Then in 1971, Engvall and Perlman, Van Wieman and Squavers published first ELISA in 1971. In 1975, Kohler and Milstein developed hybridoma technology. In 1980s, ELISA adopted for HIV, hepatitis, and pregnancy diagnosis. That's the applications. And today is widely used for any immunoassay globally is high throughput format. Giving high throughput format means you can quantify or detect protein in large number of samples. What are the basic immunological principle? ELISA exploit the highly specific binding between antibody and its antigen. A target molecule's antigen is immobilized on a solid surface, that is on macroplate, then detected by antibodies, which is linked with enzyme that convert a substrate into a measurable color product. That's important. Means the enzymes is linked with primary antibody or antibody, and this is going to be convert a substrate when we add a substrate into a measurable color product. Means you can measure the color product. Antigen coating of the plate, then we have a blocking, then antibody binding, then we have a enzyme conjugate, substrate addition, and signal rate. This is the steps for ELISA. Antibody and antigen binding is highly specific. Means that's why it is very significant to identify even a small amount of protein, peptide in a biological samples. ELISA can detect a single target even in a complex biological system. That is very unique features of ELISA. Enzyme amplification, what it does, each enzyme molecule convert thousands of substrate molecule, and that's why it has high sensitivity. And absorbance measured by spectrophotometer at typically we have range 450 nanometer is proportional to analyze the concentrations, particular target proteins or peptides. Key components and reagent, macroplate. It is 96 well polystyrene plate with high protein binding capacity. It has high protein binding capacity. and it has a flat bottom for absorbance readings and half area plate for the low volume, because we require to save the amount that's a low volume because of limitation on the samples. Capture antibody coated on a plate surface binds and immobilize the antigen from the samples, must be highly specific to the target. That is very specific. It means that we use monoclonal antibody in this case. Antigen or samples. Target analyte is a standard or unknown samples or serum, plasma, cell lysate, urine, cerebrospinal fluid, CSF, or tissue culture supernatant. These are the antigen or samples we can use to detect the protein samples with the help of ELISA. This is the typical ELISA 96 well plates. We perform ELISA in a triplicate for 96 plate. Key component and reagent and detection antibody bind to a different epitope of the antigen in the case of sandwich, or same antigen conjugated to enzyme or biotin. Enzyme conjugate we use in this case HRP, that is horse radish peroxidase, or AP, alkaline phosphate linked to secondary antibody, or streptavidin. What the substrate we use, that is TMB, that is called 3,3,5,5 tetramethylbenzidine, that is TMB, tetramethylbenzidine for HRP, and it gives blue colors, substrate for HRP. And for alkaline phosphatase we used phosphonitrophenyl phosphate for AP, and its give color is yellow. And when we stop solution, which turn in blue, it stops solution turn blue to yellow. This is the sulfuric acid. We add this, so we stop the color from blue to yellow, change the yellow. Wash buffer we use PBS twin 20, that's called PBST or TBS twin, and it removes the unbound antibodies and reduce the background signal. That is very important. To get the specificity, we need to minimize the background signal. Types of ELISA: direct ELISA, indirect ELISA, sandwich ELISA, competitive ELISA. The types of ELISA is antigen. In the case of direct ELISA, antigen coated on the plate, means in the case of direct ELISA, first antigen coated on a plate, then you need to add enzyme directly on that antigen to detect directly by enzyme conjugated antibodies, primary antibody. It is fast but less sensitive. Sensitivity is less. Now it comes to indirect ELISA. What in this case? Antibody detected by unlabeled primary enzyme conjugated secondary antibody. In this case, antibody detected by unlabeled primary plus enzyme conjugated secondary antibody, and it is more sensitive but flexibility. Now the sandwich ELISA, what it does? Capture antibody and detection antibody. In this case, capture antibody and also detection antibody, and it is highest in specificity and best for samples from like serum blood samples. What in the competitive ELISA? This is labeled and unlabeled antigen compete for a limited amount of antibody, means antibody is limited in this case, and it is ideal for small molecules or haptens, small antigens, haptens. Now for the direct ELISA principle and protocol, first we coat the plate. What it does, in this case, antigen is coating buffer, means in the coating buffer is carbonate, bicarbonate, and the pH is 9.6, added to wells, then it incubate overnight for 4 degree. So means that first We coat the plate. Now next we block the plate. What it does? Block with 1.5% of BSCA or bovine serum albumin or milk for 1 to 2 hours at room temperature. What it does? It prevents non-specific binding. That means you need to block the wells with BCA. Now we have to add enzyme and tagged antibody. The enzyme conjugated primary antibody added directly to the wells and incubate 1 to 2 hours at room temperature. What happened? Then we add substrate, that is substrate is TMB or other substrate, incubate 15 to 30 minutes in dark. That's very important. We cannot expose to the light, and then blue color develop. The blue color is developed. Now when we do that, then we stop this blue color and read. We add stop solution, that is sulfuric acid. We add hydrogen sulfate and its colors become yellow and read the absorbance at 450 nanometer. This is the total protocol for direct ELISA. See, this is a direct ELISA. Sample is added to the wells, and this is our target samples. Protein binding to the plastic, they bind to the wells. Now we add conjugated detection antibody binds to the immobilized analyte. This is the antibody bind to the immobilized analyte. Now it has enzyme conjugate. This is enzyme conjugate. This is stand for enzyme conjugate. And what happened? Then we have enzymatic color reaction. It is proportional to the bond antibody. Now we add the substrate and it is give the color, and this color is a proportional to the bond antibody. Detect the concentration of the protein. Now in direct ELISA, principle and protocol. First, we do antigen coating. Coat wells with purified antigen or sample containing antigen overnight for 4 degree. Not be blocked free site with BCA milk for 1 hour at room temperature and wash 3 times with PBST. Now we add primary antibody, unlabeled primary antibody, patient serums or specific IG for 1 hour, 1 to 2 hours at room temperature. Then we add secondary antibody, add enzyme conjugated anti-C secondary antibody 1 hour at room temperature and wash 3 to 5 times. Now further we add substrate plus we perform read, add TMB substrate and it's become blue color. Now we have to add stop solution, hydrogen sulfate, and it's become yellow. Now we read at 450 nanometer and we will get the concentration of the protein. Sandwich ELISA is a gold standard for protein quantifications. Means when we quantify, we need to do sandwich ELISA. Most widely used format for cytokines, hormones, biomarker quantification due to exceptional specificity at very unique and special specificity. Coat plate with capture antibody blocked unoccupied site. Add samples, that is antigen binds, then add detection antibody. Then we add conjugate or enzyme conjugate or streptidine antibody. Then we add substrate and get the color signal. This is indirect ELISA. Protein coated container, then antibodies bind to the protein. This antibody bind to the protein. Then excess wash away. Then we add secondary antibody and which bind to the first is become sandwich. And indirect ELISA. Then further we second antibody bind to the first. Then we have a visual changes confirm the correct protein is present when we add the substrate. Enzyme is attached with secondary antibody. When we add, its color is going to change. Sandwich ELISA. The requirement, it's required 2 antibodies that bind to the non-overlapping epitope on the same antigen-antibody pair. In this case, antibody pair form is, and capture antibody typically coded at 1 to 4 macrogram per ml in carbonate buffer overnight at 4 degree. The reduction antibody can be biotinylated or streptavidin HRV for enhanced signals. And it is excellent sandwich ELISA for cytokines study expression like interleukin, TNF, IFN, and hormones like thyroid stimulating hormone, insulin, and growth factors. A standard curve we draw usually for 7 to 8 points enables accurate quantification of unknown. This case, accurate quantification of unknown. Now, this is sandwich ELISA. Wells are pre-coded with capture antibody. This is antibody already we have pre-coded. Now we add the sample. Sample is added and analyte is bound to the capture antibody. This is a samples capture antibody binding. Then we added conjugated detection antibody bind to the immobilized analyte. So this is the immobilized analyte. Then we have a secondary antibody that is conjugated with substrate or fluorescence, whatever we have analyte. Then what we have in direct detections and enzymatic reaction is proportional to the analyte. So whatever the amount of color of the solution, that is giving you idea that it is directly proportional to the analyte. And it looks the sandwich, first antibody, second antibody, that's why it's named sandwich ELISA. And these are the untargeted sample matrix. Now the competitive ELISA, it is inversely proportional signal and it is ideal for small molecules. What is the principle of competitions? Unlabeled antigen in the samples compete with the fixed amount of labeled enzyme conjugated antigen for a limited number of antibody binding sites. Higher antigen in the samples, it has less signal detections. What the application? It is a small molecule analytes identified like steroids, drug of abuse, pesticide, vitamins, haptens. Hormones like cortisol, estradiol, testosterone, progesterone, and it cannot differentiate between structurally similar molecules without careful validations. That's a limitation of competitive ELISA. Signal relationship, sample, none are low, the competition is minimal, signal output is high, maximum OD, and the interpretation low antigen concentrations. When we have a sample as a medium, competition is partial, then we have a medium OD and we have a medium antigen concentrations. When the sample antigen is high, we have competition maximum and OD is minimum, but a high antigen concentration is indicating. This is competitive ELISA. Wells are pre-coated with secondary capture antibodies. Secondary capture antibody. So the capture antibody conjugated and samples are added simultaneously. Both conjugated and samples are added together. This is conjugated and samples are added together. Then analyte and conjugate compete for binding. Then analytes and conjugates compete for the bindings. Then what happened? Enzymatic color reaction is also proportional to the bond conjugate. That's also same condition. The enzymatic reactions, then we have a color of the product and that is proportional to the bond conjugates. Materials and equipment. Plate reader, we need macro plate, a spectrophotometer, and we have a two substrate that is TMB 450 nanometer and PNNP at 405 nanometer, and we have multi-mode reader prefer, and we need plate washer that is automated 96 plate washer or multi-channel pipette for manual washing. Then the temperature of the incubator and shaker should be controlled and plate shaker are room temperature 20, 25 or 37 for incubation of antibody. Materials and equipments, we need pipettes, single or multi-channel pipettes, 8 to 12 channels, or for consistent dispensation of the solution in the 96 well plates. Then high binding polystyrene, 96 well supposed to be high binding polystyrene flat or half area wells brand consistent important. Then we need ELISA kit or antibody, match antibody pairs or commercially ELISA kit with all reagent included. Also require coating buffer, that is carbonated pH 9.6. We need wash buffer, that is PVST. We need blocking buffer. We need stop solutions, 2N hydrogen sulfate. Then we distill water, timer and lab notebook to take the readings and also the protocols, write the protocols. Sample preparations. Serum and plasma, we collect the blood, allow clotting of the serum or use anticoagulant in the plasma and then centrifuge 1000 to 2000 G and then aliquot and freeze the sample at 80 degree and avoid repeated freeze and thaw. This is very important because it degrades the proteins. Then from the cell lysate, we add the cell lysate with RIPA or NP buffer and then protease inhibitor. Then we centrifuge the samples at 12,000 G for 15 minutes at 4 degree and quantify the protein with BCA assay. We can also take samples from urine, centrifuge to remove the cells, use fresh or freeze at -20, that is fine for the protein. We can keep for 1 and 2 days or maybe week. May need concentration or dilution depending on the analyte. Then we have a tissue extracts, homogenize the tissue in PBS and we add the protease inhibitors and the centrifuge, use supernatant and normalize to the total protein content. Step by step protocol. Day 1, first step, coat the plate, dilute capture antibody in coating buffer, that is we have the concentration 100 macromolar per well, seal the plate and we incubate overnight at 4 degree. Day 2 and step 2, we wash and block. Wash the plate 3 times with PBST. Then we add blocking buffer, that is 200 macroliter per well for 1 hour at room temperature on a shaker. Then day 2 or 3 steps, all standard and the samples. Prepare a standard curve, that is 2-fold dilutions. Add 100 macroliter per well and 2 hours at room temperature or overnight at 4 degrees centigrades. Day 2, step 4, detection antibody. Wash 3 times to remove the non-specific binding. Then add biotinylated detection antibody 1 hour at room temperature. Or we can add, then we have to add streptavidin HRP and then we add wash 3 times. Then we add SA-HRP conjugate and 20-30 minutes room temperature and protect from the light. Then we add TMB substrate. Then we wash 5 times and add 100 macroliter of TMB substrate and further incubate 15 to 20 minutes in dark, then the blue color develop. Blocking and washing step is a general purpose, works well for most target preferred by phosphorylated protein. When we use 1.5 to 1% to 5% BCA in PBS, or when we use 1 to 5% non-fat milk, it is cost effective and for the most of ELISA and avoid with a biotin avidin system because milk has a biotin. So when we use any kind of biotin, then avoid with biotin avidin system, we not use the non-fat milk. Also, there's a commercial blocker, casein, fish gelatin, super block, optimized for a specific assay type and is higher very cost. Blocking and washing step. In washing, we use PVST, PVS plus 0.1 twin 20. Twin detergent disturb the non-specific hydrophobic interaction. Is the role of twin detergent, excuse me. And it's a standard, we have to 3 to 5 wash for 5 minutes each with 200 to 300 microliter per well. Fully aspirate between each wash. Insufficient washing giving the high background, and overwashing lowers the signals from disturbed specific binding. Then we use automated plate washer, ensure consistency across 96 well plate and is highly recommended for the ELISA. The final was some protocols use PBS without twin to avoid detergent interferences with the substrate and we get high noise. Antibody incubations, capture antibody, concentration should be 1 to 4 macrogram per ml and we use buffer carbonate buffer as pH 19.6 and the time we use mainly overnight at 4 degree and volume is 100 microliter per well. Detection antibody in the case of sandwich, we use concentration 0.1 to 1 macrogram per ml. Maybe biotinylated time is 1 hour for room temperature and optimize air per data sheet. Antibody incubations, secondary antibody we use like with HRP. Dilution 1 to 1000 to 110,000. HRP or AP conjugated. Time is 30 to 60 minutes at room temperature and incubate in the dark. It's very important to protect from the light for better sensitivity and result. The primary antibody in the case of indirect dilution 100 to 110,000. Buffer we use blocking buffer. Time we use 1 to 2 hours at room temperature or overnight. Volume is 100 microliter per well. Substrate signals and detections. When we use enzyme HRP, the substrate is TMB. TMB stands for 3355 tetramethylbenzidine and it's give the color blue color and we measure at 450 nanometer with a spectrophotometer or ELISA reader. We use ELISA reader most common. This is the most common substrate, high sensitivity and reaction is stopped by hydrogen sulfate. In the case of alkaline phosphatase, we use BCIP or NVT. This is BCI stand for 5-bromo 4-chloro 3-indolediol phosphate and it gives blue color, purple blue color and we measure at 570 nanometer used in membrane based or colorimetric ELISA variant. We use for membrane based or colorimetric ELISA variants. Mainly we use HRP conjugates. What are the application of ELISA? In the case of infectious disease diagnosis, we use lot for ELISA for HIV detection of antibody, HIV antibody, human immunodeficiency virus, HPV or SCV antibody detection. Also, we use ELISA in for the SARS-CoV COVID viral serology testing, COVID virus, SARS-CoV virus serology testing. Then also we use malaria antigen diagnosis and Lyme disease confirmation. So these are the routine for detection, routine techniques for a testing or for a detection of different disease in the hospital or in the lab. Then also for autoimmune disease, like to detect the rheumatoid factors detection or any select disease like we use anti-TDG antibody and also for thyroid we use thyroid antibody for detection of thyroid hormones. Then for oncology we use like different markers, tumor markers like CA120, PCA we can use for oncology markers. Then also we use cytokine profiling in TME Then also we use angiogenesis factor like VEGF, vascular metabolic growth factors, and also we monitor the drug concentration. When we use to monitor the drug concentrations, we use with ELISA. And also it has very important for the drug development to know the particular novel drug pharmacokinetic studies, and also we use anti-drug antibody detections, and also important biomarker qualification, and also therapeutic drug monitoring, that how this particular drugs target the particular specific proteins, and then we can have to check the ELISA, the concentration of the particular protein. And also used for food safety when we wanted to check the allergens. So we use in the case of food like peanut allergen, the gluten, this is according allergy, that's for food safety. Then also we check the antibiotics residue in the milk, if any antibiotics residue present in the milk, which used for check the with ELISA. Then also we use mycotoxin screening with the help of ELISA, and also we check genetically modified protein in the any food items or any grains or any feed, genetically modified protein products. Then also it has lot of application in endocrinology, in hormone quantification like TSH and LH, thyroid stimulating hormones. Then also we have to check the insulin, C peptide assay. Then we can check the expression, quantify the cortisol, ACTGS measurement, pregnancy test. We have beta HCG hormones. We check with the pregnancy check. Apply the ELISA. The summary of my lecture: ELISA uses antigen antibody specificity. That's very important. For it is a specifically antigen and antibody specificity, and we have enzyme amplification for sensitivity of protein detections, and mainly four formats, that is direct, indirect, sandwich is most common. Then we have a competitive ELISA. Sandwich ELISA offers best specificity and sensitivity protein quantification. This is the best one. And a standard curve with R square more than or equal to 0.9 required for valid quantifications. And we have a control, three control. We use positive, negative, and the blank. Then we duplicate, and percentage of monitoring are very important. Significantly, we can check this percentage. CV percentage monitoring is very important and essential. An application is span diagnosis, drug discovery, food safety, and basic research. So there are very multiple importance for this ELISA. References I've taken for this lecture: Engvall, E. Perlman, 1971, Immunochemistry, first ELISA paper. Then Van V. Man and V. Kescom, 1971, Phelps letter, concurrent ELISA development. Then Lipkin, RM, clinical chemistry, historical overview of ELISA. Then Soleimani S. and Mani M. is 2018, Biosense Bioelectron Modern ELISA development. Thank you so much for listening my lecture. We will meet next lecture. Thank you so much.